This thing is rather slow today, so we'll see how it goes. All right, so our next unit is 6B. I know we went from 6A to 5 and then to 6B. It's just a long story, but short of it, I think it's better to do it this way. Sequence-wise, I think it's better for you all. So we were about meiosis, and we're talking about genetics, and we're talking about how we can use these Punnett squares to determine what offspring can look like. But the question is, okay, great, so they can look like this, but how is it they actually get to look like that? How is it that we have this DNA that you inherit, and how do we take that DNA that you inherited and we turn that into what you actually are? Okay, and that is the story or the process of what we call gene expression. So we're going to take that DNA and we're going to turn it into a product that produces, in general, a protein. So the information content of genes is the specific sequence of nucleotides. We talked about, DNA has a phosphate base, or has a sugar, a phosphate, and a nitrogen base. And those nitrogen bases are different: A's, T's, T's, and G's. That's the sequence of the nucleotides. Okay, the rest of it's the same. Sugars are the same, phosphates are the same. The DNA that is inherited will lead to specific traits that are dictated by the synthesis of proteins. Proteins are the links between genotypes and phenotypes. So you have a particular genotype, that's a particular code, that's a particular recipe. And how do we turn it into a phenotype is what gene expression is. Okay, so we're going to take that DNA and we're going to turn it into a protein, and that protein will then be our phenotype. That will be your skin color, or that will be your eye color, or that will be your resistance to certain diseases, or your lactose intolerance. And we're going to talk about that in terms of two things: transcription and translation. Who remembers transcription and translation from freshman bio? Maybe. Okay. Don't worry. We're going to go through it. Okay. Let's go down to this next one. Not all proteins are enzymes. Many enzymes are proteins. Most enzymes actually are proteins. But there's a hypothesis of this thing called one gene, one outcome. And at first it was one gene is an enzyme, and now we think we make it more general, and we say it's not just an enzyme, it's a protein, because not all proteins are enzymes. Many proteins are composed of several polypeptides. Each has its own gene, which is kind of cool. So if you've got, remember we had primary, secondary, tertiary, and quaternary structure for proteins. So we had first level, second, third, and then some of them had a fourth if you had more than one chain. So we now know that those proteins, those chains, are oftentimes, they have their own gene. So one chain and another chain will have their own DNA that will then combine to produce a multi-stranded or multi-peptide protein. I'm going to skip through some of this because it's not really that relevant to us. Let's see what this is. Okay, so basic ideas and words that you need to know. So DNA we've already talked about, right? Deoxyribonucleic acid, we know. But now we're going to be bringing another one in here that is RNA. So RNA is ribonucleic acid. And what was RNA? How is RNA different from DNA? It's one-stranded. Good. And what else? There's one other thing. There's kind of two big things. Uracil. You're going to use U. You're going to use that U instead of, we're not going to have, it's going to replace T. T is not going to be there in RNA. It also has ribose for sugar rather than deoxyribose. But somehow we have to go from the DNA that's stored in the nucleus to getting, where do we make proteins? Where do we make proteins? In the ribosomes. So we're going to have to get that DNA code some way, somehow. We've got to get that to the ribosomes. But before we do that, well, let me step back and say, DNA is always in the nucleus. It doesn't move. It doesn't leave. And the idea is that it's too important. If it left the nucleus, it could get lost. It could get lost. It could get broken. We want to keep it in one location, okay, like a master location. So we want to keep that in one place. So there has to be something to get it to the ribosome, get that message to the ribosome. And to get it there, we're going to make a copy of it. We're going to make a copy of it with this molecule, or with this process called transcription. And the molecule that's going to be responsible for doing that is going to be our mRNA, or messenger RNA. So M meaning messenger, it's making a copy of it, and it's taking that message out to the ribosome. And then once it's in the ribosome, that code, I mean, it's just like any code, you need to change that into an actual protein. So in the ribosome, our last step is going to happen, or our second step is going to happen, which is translation. The translation is I take my mRNA and I turn that into an actual polypeptide chain that will in turn become a protein. Okay. In prokaryotes, there is no nucleus, right? So that, as we will see later on, actually makes transcription and translation happen pretty quickly, because they're not as sophisticated as we are when it comes to protein synthesis. So they're going to do a little less than the eukaryotes are, and transcription happens, and then translation happens before transcription is even done. So transcription's starting, it's getting all the way, might be half the way done, and translation of that first part that's been transcribed will happen. So it happens very quickly, and we'll talk a little bit more about that as we go. In eukaryotes, though, there's a nucleus. There's a nuclear envelope that physically separates, and acts kind of as a barrier to transcription and translation. So one can't happen right after the other. It has to get through that nucleus and through that nuclear envelope. And so we'll talk about that, because in eukaryotic cells, there's going to be some modification that happens. And that modification is going to be important in determining what kind of mRNA message you get. And just because you get one code when you start transcription doesn't mean you're going to end up with that. You may end up with a slightly different message. So here is an example of a bacteria cell. So transcription happens very quickly, followed also very quickly by translation. No nucleus. It's all in the cytoplasm. Did you find it? Is it just on the next page? No? Can't find it? No está aquí? How about that one? Any of those? Kind of only counts in horseshoes and hand grenades. That's not what I wanted. All right, I just opened up what I just printed. So why isn't it there? Okay. Well, that's a problem. Just listen. So the first page was okay, right? You know what? I might have taken some of these out. No, I didn't though. I don't know. All right, so just look at the pictures. Look at the pretty pictures. In eukaryotic cells, you have a nucleus. So the first thing that's going to happen is this transcription. So let me go back up to the last one because you don't have it. So this is in a bacteria cell. And there's just a cytoplasm. There's no nucleus. So transcription and translation happen one right after the other. But when you get to, I'm just going to show you this one down here because it's a bunch of them. But when you get to eukaryotic cells, transcription happens. And then there's some processing that happens. You have to edit the RNA, before it can leave the nucleus. It's like a kind of a checker that's going to go on there, and there's going to be some processing that happens. And then it's going to have to go through the nucleus, and it'll go into translation. So there's just an extra step there that has to happen, and we'll talk about all of that. You have this. This is so weird.
Where is that on the slide?
We have 12.
Guys,
We have 13.
But you have this, right?
Yeah, that's where we start.
What I'm worried about is I wonder if it copied every other page, and I don't know why it would do that.
Nick has it.
I do.
You're the only one who has it. Let me see your staple.
What can I say?
Let me see your staple. Yeah, I know. Yep, you got the original. So that's what it did.
Yeah, the original.
I'm still confused.
Oh, that's a nice.
All right, so I'm just going to do one more page, and then I'm going to stop. So this one was just kind of not a lot of information. So back up here in this previous picture, we have transcription, RNA processing, and translation. So that transcription, that is called the primary transcript, meaning that there is an initial RNA that's generated, copying the DNA. But then before it can actually go on, there has to be some processing that happens. Okay? And that's what I was showing above here, this processing. Okay. I'm going to go on to here. So the question is, how are the instructions for assembling amino acids into proteins encoded in DNA? So let's step back there for a second and just remember, what are proteins made of?
Amino acids, bro.
They're made of amino acids. Okay? So there's 20 different amino acids. But there's only 4 different nucleotide bases, A, C, T, and G. Well, yeah. What's that? How do they do that? So the question is, how many letters would you need to come up with enough codes for 20 amino acids? Well, I don't know. A lot of them. We're not going to worry too much about that. Let me see if social distancing. Thank you. This one. So it turns out that to make every amino acid, you need to have 3 letters. So we call it a triplet code. So every amino acid is going to be something like ACG or GGG or GAU, okay, or GAA. And that's the code that we have to have. If you have that triplet, That's enough to code for all of the amino acids. So if I start with a DNA template and I transcribe it, and remember I replaced my T's with U's, ugh. Each 3 letters will code for an amino acid. Okay, so in this case we have 4 codons, which is 12 bases, and we have 4 amino acids produced: trip, phi, li, and gly, and sir. And we'll talk more about those later. I am going to stop because you guys are focused as much as a herd of cats right now. And